everyone, I'm Carmen, Managing Director at Hanson Wade, looking after the Process Development and Manufacturing series. Ahead of our Viral Vector Process Development Summit, which is taking place in January in Boston, I'm very excited to be catching up with Michael Mercaldi, who is the Vice President of CMC Purification and Drug Products from Oxford Biomedica Solutions today, to get his perspective of why viral vector is going to be so important and critical to our cell and gene therapy in the near future. Hi, Michael, how are you?
Hi, Carmen. How are you? I'm doing well. Thank you.
Thank you so much for spending some time with me today, and super excited to be working with you at this meeting. Also, Michael, I know you've got a lot of experience in the AAV arena, but would you mind perhaps giving us a little bit of a highlight on your journey in your career so far?
Sure. Yeah, I'd be happy to. And thanks for the nice introduction. So as you stated, I'm right now, I lead the CMC program management, purification, drug product teams here at Oxford Biomedica Solutions. I came to Oxford Biomedica Solutions through homology medicines. So to give you a background, Oxford Biomedica Solutions was formed between a joint venture between Homology Medicines and Oxford Biomedica based in the UK. And I was on that team for almost 5 years at Homology Medicines, where we helped develop our best-in-class AAV platform. Where I was leading the purification and drug product function. Before that, I had roles of increasing responsibility in the purification process development space at MedImmune AstraZeneca, Merrimack Pharmaceuticals, Kodiak Biosciences. My education is I have a bachelor's in chemical engineering from Rensselaer Polytechnic Institute and a PhD in biochemical engineering from Tufts University.
Thank you for the nice introduction. And certainly you have been in this space for a long time, and also switching from a drug developer perspective to now a CDMO perspective.
Yeah.
Obviously, viral vector is not something super new, but we've seen a lot of changes over the last 12 months, loads of investment, loads of GMP manufacturing. And I think everyone in the industry is right now looking into the process development optimization. But we'll be keen on getting your perspective, how you see this space has evolved over the last 12 months, and perhaps what is the priority for us in 2023?
Yeah, no, that's a great question. And this is an interesting time, I think, with all the market turbulence that we're seeing. So what we've noticed in the past year was that a lot of product developers are really focusing on product quality, AAV costs, and funding challenges. Which is actually a very acute need for a lot of drug developers right now. And we kind of see these themes as really interconnected, and we feel that we can help our customers address them. More specifically, we are seeing challenges in the funding space for drug developer companies due to the rising interest rates, but also as a reflection of some of the safety results and uncertain return on investment of some of these cell and gene therapies as well. Investors are starting to be more cautious with funding and are demanding more robust data packages before they decide to invest in drug developer companies. And this is where we think we can help because the funding's not as significant as it was years ago. So the drug developer companies may have to look at ways to manage their capital more effectively and in a more focused manner while balancing their kind of CMC drug development needs with their clinical program as well. So we think that by working with us, we believe that we can help drug developers overcome these challenges. And instead of choosing to build out internal AAV CMC capability, they can which could take years and significant investment, significant capital, they can actually choose to work with us where we have all this built out and at their disposal. And we feel that with this capability that we offer, that we can actually help them balance their speed to the clinic and speed to commercialization while managing their cost effectively and then focusing on the clinical execution at the same time.
Absolutely. I think 2022 was a challenging year for quite a few gene therapy drug developers because of clinical hold. And also, I think the last 2 months we've seen some confidence re-injecting into the space, seeing the FDA approving. But from some of the conversation, a lot of people are actually saying because of the clinical hold, we now need to spend more time and investment into the process development and CMC. What's the correlation there? Just keen on understanding your perspective of why process development is now even more important to support the candidate through the clinical development phase.
Yeah, no, that's another great question, and that's true. There have been some issues in the clinic. People think it's product quality related. Could also be drug design related as well too, where we think product process development can help is more on the product quality side, and that's what we can offer as well. There are the biggest kind of impurity right now that people are trying to control is empty capsids. And that's where our platform, we've effectively reduced our empty capsids to levels less than 10%. But there are some drugs in the clinic right now that are going in with a lot more. And that's really driven by regulatory risk, appetite, and everything like that. But we kind of view those as an impurity that needs to be controlled and removed, and that could have a negative adverse event, because you're just injecting more foreign material into your patients. Outside of capsids too, it comes down to the construct design as well. Some of the construct designs may not be as effective, which necessitates extremely high doses, right? And that's something that we can also help out with too, where we have an experienced team of vector engineering scientists who know how to design these vectors so that they can express better in vivo, they can deliver better in vivo, and then we can reduce the overall doses. And reduce the overall foreign matter being injected into patients, and then ultimately help mitigate these safety events that have been occurring. So that's where process development kind of all fits in, is that we can help with the design of the vector, and we also help control the product quality as well too.
So I think like that's part of the conversation that I've been hearing, and that's kind of like one of the mission what we will try to bring to the table in January, how we can actually reduce the impurity level, make sure we've got a robust CQA when we're thinking about the process development, and that will support efficient scale up. Obviously, there are loads of CDMO in this space, so really keen on understanding like what are the common pitfalls that you've observed from drug developers when they come to Oxford Biomedica, and perhaps like how you guys differentiate from other peers in this space as well?
Yeah. So I think the common pitfall that we've noticed is there is tremendous pressure on drug developer companies to move fast, right?
Yeah.
So research scientists need to get that proof of concept very quickly. And then as soon as they get that proof of concept, nominate that candidate, go in the clinic, go. What we actually advocate for is to kind of take a little bit more cautious time during that construct development phase. And then we actually can help customers and clients get to the commercialization faster and get their proof of concept data faster, because it all starts with that construct development phase. If that construct is not maybe the best construct in vivo, or has manufacturing liabilities, or if it's not the best expressing in vivo, but it kind of works and good enough to go, then you're going to have some issues later on that you're going to have to deal with. And by the time you're in the clinic, you're moving towards commercialization, it's very hard to change any of those attributes. And that's where we can step in, is that we kind of focus on speed to success instead of speed to clinic, where we can work with you to help effectively design a construct, leverage our very high performing manufacturing platform. And then we can get you into the clinic in a reasonable amount of time, but then everything else on the back end, going into your later phase development, going towards commercialization, is just going to all fall in place with minimal headaches, and going to really ensure your success for commercialization of your therapies.
So it seems like you will be the trusted partner from start to finish, and hopefully like that will eliminate a lot of like back and forth tech transfer, and you can actually work with them throughout the early discovery, the process development, all the way to commercial manufacturing phase. So it's a lot more seamless.
Yeah, correct. I think that's a really nice way of putting it. Where we think we have a nice value that we can offer to customers and clients that other CDMOs may not offer is really through that construct design vector engineering service, and then having this really experienced team of drug developers that we're not just taking a process and doing manufacturing capability, we're really an end-to-end solutions provider to help develop products. And that's really what our mission is and what we really love to do.
Good to hear. But of like looking into in a month's time, we will have you speaking on behalf of Oxford Biomedica on something super interesting, particularly on the bioreactor. Would you mind giving us a little bit of a snippet or heads up in terms of like what that would encompass, what we can look forward to?
Yeah, I would be happy to. So what I'll talk about today is kind of introducing our organization, our intent capability, but more specifically in the technical side, we've had a lot of great success this year. We are now able to achieve across multiple constructs and multiple serotypes a bioreactor titer of 1E15 VGs per liter, which we think is one of the best in the space right now. We also are able to deliver less than 10% empty capsids and 90% full vector in our drug substance as well. So I'll kind of talk about how we've achieved those things and how we've effectively scaled them up as well, and kind of our strategy towards scaling it up to ensure that just because it works at PD, it definitely works in manufacturing as well.
Absolutely. We've been doing some conversation or conducting some research with the European viral vector drug developers, and everyone just said like the yield is their biggest headache at the moment. even though they can achieve commercial manufacturing, the yield is not quite there. And without
Yeah,
the yield, you just can't reduce the cost of goods. So look forward to your presentation with a lot more technical details in January.
of course.
On the flip side, what would be your biggest question or burning question you'd like to get answered, or perhaps like throw it to the audience so that we can get them thinking before coming to the meeting in Boston?
Yeah, I think the question, I was kind of thinking about this for a little while, and I think it kind of brings together a lot of the themes that we've been talking about and I've been kind of hearing in the space is, what can we as process developers do to improve the safety, efficacy, and accessibility of cell and gene therapies?
That's a very good question. Particularly, I guess right now the cell and gene therapy is still very, very expensive. So if we can actually do our parts to improve the process development, the scale up, it will eventually reduce the cost of good, and that will actually help bring the curative treatment to patients with affordability and accessibility.
Correct. Yeah, and that's what we hope to do. And I think with my talk of the high titers, that's a step in that direction because we can definitely make a lot more vector and really increase the value proposition where we can really reduce those cost of goods for drug developers quite significantly with their highly productive process.
Thank you so much, Mike, for sharing your thoughts. And definitely, we look forward to working with you and seeing your presentation come to life in January. Um, I hope we can also do our own little part to help the AAV community to achieve scalability, reduce impurity level, and obviously it's always about like the product quality and safety to our patients. So thank you so much for today, and I will see you in Boston in January.
Okay, great. Thanks, Carmen. Appreciate the time, and I'm looking forward to seeing you in person as well.